Hi, welcome to the Anti-Waffle podcast with me, Darcy Naylor, and Marvel Gregorio de Souza. Hello! And together we'll try to talk about research without any gobbledygook. Each episode we'll be talking to a researcher who will discuss the work they're doing, looking at why this research is important, and asking how did they get into this field. We hope this will be informative and interesting and bring a bit of the academic world into the real world. This week we have Dr. Chris Smith at Queen Mary's University, London. Hi, Chris.
Hiya.
Hi. Thank you very much for being here.
No problem. Thanks for having me.
Thank you. Would you like to start by telling us a bit about your research, please?
Yeah, sure. So my research basically is focused on the different wound healing between the gums and the skin. As people may know, your gums heal much faster than the skin. They don't scar as much. So if you have a tooth removed in dental surgery, they won't stitch it up, they'll just leave it. Whereas obviously compared to skin, you'll have stitches and you'll have lasting scars. So my research was focused on understanding why there's that difference between the two tissues.
When did you start looking into your research?
So I started my PhD back in 2014. I actually only just finished a couple of months ago and got my doctorate last week.
Yay.
Which is very exciting.
Yay!
So my background, I just did biology at York, just a straight degree, and then did a master's in biology as well. It was all very focused, actually it was quite focused on parasitology, and that's what I wanted to go into. But as anyone who's applied for PhDs will know, it's quite hard to always get the PhD in what you want, especially if what you want to do is neglected tropical diseases, because the clue's in the name. So I decided I want to stay in health and disease, and a PhD came up, which was looking at scarring and wound healing. So I decided to go for that. I was quite lucky that I was partially funded by GSK as well. So like a case studentship, so I had the opportunity to work with both academia and industry throughout the 4 years, which I think is really useful for anyone who's applying for a PhD to look into that opportunity to work with academia and industry.
I always assumed that they just left it kind of like open in your mouth with the gums because it was in your mouth and there was less chance of infection.
There's several reasons that why the gums heal faster, and most of them we didn't look into. So I'll talk about what there is normally. So saliva, as everyone probably thinks straight away, that does play a big factor.
Right.
So your saliva has lots of growth factors and antimicrobials that kill the infection in your mouth and allow the tissue to grow as well. But then on top of that, your tissue reacts very differently to your gums react very differently to skin. You have a similar number of blood vessels between the tissues, but upon wounding, you'll get a lot more blood vessels in skin than you will in gingiva, and that delays the whole wound healing response and it prolongs inflammation, and that's what leads to the scarring. And also the fact that you have more blood vessels leads to more white blood cells, which also prolongs the inflammation. But then my research specifically was looking at the different gene expression of certain cells. So I found that the epithelial cells of the tissues express different genes that are involved in wound healing quite differently. So I was looking at a very niche part of the wound healing and putting it all together with what's already known. The outcome is that we found specific genes that are upregulated in the gums compared to the skin that have a role to do with wound healing. Annoyingly, as I'm sure people who listen to this will also have a similar experience, I was about to publish my research and someone else scooped me and released all there was 4 genes that we were particularly interested in, and that all got published the week before I submitted my thesis. So the long-term effects of part of my project are a bit redundant now. This is someone else already looking into it. But hopefully the idea is that we can improve skin healing to get it to a similar level as the gingiva healings, whether that's reducing scarring or just healing the wound rate. So a lot of scarring actually comes from surgery rather than bumps and grazes. So there could be a possibility of, in surgery, trying, if you have certain ointments or creams that increase the expression of some of the genes that we found, maybe that could increase that. Yeah, genes expression was increased, then that could potentially improve the healing.
So if other people are looking into this research too, is there not opportunities for you to collaborate with them?
Well, the thing is as well, like I say, I've just finished my PhD and the funding ran out. My lab that I was in has closed. Right. So ideally, there would have been a good opportunity. They were a lab in America, published in Science, which was a bit above me, so it would have been quite nice to work with them. And maybe I could reach out to them, but I'm at a bit of a fork in my career at the moment, and I'm thinking of leaving the field and going into something else, which could be adrenal glands, or it could be working in industry. But there's just a few of the options I've got at the moment.
Right. So it must be, looking at that, still staying on the subject of that kind of frustration where you've been working on something and then someone else, because like I write scripts, and so I have had cases where I've written something and then the film has come out. Do you think there is an answer for that? Do you think it should be like maybe research should just come from one place, like internationally, and that
well, you have different sections working on different parts.
Maybe you have to get like a license to do research so that there is only one person, or do you think that competition is healthy?
I think competition's healthy. I think that it brings different perspectives to it. So, and people have different ways of looking at things. So, a large part of my project was building 3D organotypic models. Actually, that was the original start of the project, and it just kind of changed. 3D organotypic models is basically a way to reduce animal testing. So you grow 3D models of human skin or human gums, and then I wounded them, and I showed that my gingival models were healing faster than my skin models. So that's all very in line with 3 R's of animal testing, which I don't know if you know, but they're replace, reduce, and refine. So it's all a way of trying to take animal testing away and use all these different in vitro models, which is basically what my project was on. And then it got moulded into wound healing. But so we were looking at that, whereas the group over in America looked at human testing, and they actually did biopsy punches on people. And they also looked at mice, which we weren't able to do. Yeah. So I think the competition is quite good because even though, yes, they looked at the specific genes, I never looked at the animal testing and they never looked at the in vitro models.
Instead of competition, could there not be collaboration? Could there not be a wider way? Because I always think there must be all this research going on. And this is part of why we're doing this podcast, is to get the people to understand that there's more research going on than you'd ever know about. But then it must be the same within universities, and I don't even know, within the same university, how does everybody know what everybody's doing? Yeah, I think
that is a massive issue. I think there are people, I heard stories of the institute that I was in where people were working on very similar stuff, just the other side of the buildings, and they didn't realise, and if they'd actually come together, they could have solved it so much faster. And I guess that's why there's conferences and all this kind of stuff, so that you can go along in similar fields and work together. But I think collaboration is key. I think it would just speed up research so much faster.
It just feels that there needs to be some sort of system somehow that everybody can feed in what the research is and what they're looking for.
Well, exactly. If you just put out a brief and say this is what we're looking at, you don't have to say what genes you found. You could just say we're looking at the difference in healing rates between this. Because the problem is there's a lot of professors out there who publish for years. You can tell they're working on it. But when people who are just finishing their postdocs, they're becoming lecturers and they're like, I want to work on this now, then there's no real
some early career pathway.
There's no news report on I'm working on this now, so just so you know, I'm going to publish this and scoop you. I think it's one of those things that could definitely be improved.
So have you had your work published?
So it's going to get published pretty soon. One of the issues actually is because the lab's closing down, there's not that much scope for much further work. And I'm sure people know getting published isn't just handing it in. You get lots of reviews back and lots of corrections to make. So that could be a bit of an issue for me. But it's something we're going to have to look into. I think, like I was just saying, I got a bit scooped, but I think the in vitro model side of my project makes it still quite niche and still quite very publishable stuff. So I think it's very interesting. So what I focused on was they're called 3D organotypic models. So they are basically, so in your skin you have 2 layers. You have your epidermis and your dermis. In your epidermis you have cells called keratinocytes, which differentiate to make your layers of the skin, as you may see in the diagram. And in the dermis you have fibroblasts and collagen and all of the kind of stuff as well. But then these models basically we seed collagen with fibroblasts, which we model the collagen, and then we add the keratinocytes on top so they can differentiate, and we have a 3D model of skin. Or gums, or people use it for esophageal tissue, and all kinds of stuff. And then you can look at different parts of skin. People are looking at plantar skin, which is the sole of your foot, as the skin is a lot thicker there. And there's all kinds of different things you can do with that. So that's what the organotypic models I work on. But then you can go further, and organ on a chip is becoming a big thing at the moment, which is basically a microfluidic system of a human. So for example, you could use human lungs, where you have two different passageways, where you have air going through one and blood cells going through the other, and you can see the cells moving in between, and it's basically just making a 3D system where you can change the levels of glucose in the blood, you can change the amount of oxygen in the air, you can look at all the things that you can look at. Well, not all the things, you can look at many things in vivo and in vitro.
You're developing, or that you could...
Yeah,
with funding,
yeah,
that you could develop,
yeah,
so that people would use it to test it, so they can check, they can change the variables on it.
Yes, exactly. Yeah, that's a better way of describing it. Yes, yeah. So I think the models I've done are very interesting, but it looks like from where the literature is going, that organ on the chip is the future at the moment. So basically, the chip is the size of a pound coin, and you can change all the variables inside it. So you can look at several different things. There's ones for skin, lungs.
So sorry, is it like a computerized?
Yeah, it looks a bit like a computer chip, but it's basically imagine like an elongated pound coin, and there's little fluidic vessel chambers inside it. And then in the middle there may be a layer of... So I'll explain the lung one. So in the middle there may be epithelial cells, and above it is just air with the amount of oxygen, carbon dioxide you can control. Below it is like plasma with blood cells in it, and then you can change the amount of glucose or whatever, or change the amount of iron in the blood, and you can analyse the difference that makes. So it looks like that's kind of the future. But then people have done it with skin and all these other things as well.
Does that sit in like a model, or
no, so
they just literally test on this little pound coin?
Yeah. I mean, I'm no expert. Someone who's a skin and chip expert, they might catch me out on this. But I think because they're also so small and they're quite, I'm going to say easy to manufacture, but I think they are. But you can manufacture them in a large quantity. You can do the very high end number for testing. You can get like a lot of data quite quickly out of them. Whereas the models I grow take probably all in all about 3 weeks to grow.
Right. But that's the beginning, isn't it?
Yeah, exactly. Yeah. So
prototype.
There's the 3R movement, the NC3R movement is a big movement to try and move away from animal testing, and there's all these different models out there to try and improve that. So it looks like it's a very exciting time to move away. Cause also, a lot of people use mice for animal testing, but mice are 97% similar, I think, to us, but the 3% genetically, but the 3% is actually a massive difference. Because I think we're 60% identical to bananas. And so you can tell, like, it's all very different. So, and as a skin scientist, you can't really use... Well, mice aren't the best model for skin. The amount of hair follicles they have is completely different to us. So using animals doesn't always necessarily translate to human.
How did you end up being a skin scientist? Did you always know that this is the sort of area you wanted to go into?
No, not at all. So, like I said, I kind of wanted to be a parasitologist originally. Sorry?
What's that?
So parasites to work on, I wanted to work on malaria and trypanosomes, African sleeping sickness and all that kind of, that's what I really wanted to work on.
What attracted you to that?
It was just, I remember in my undergrad modules, we had modules on cancer and bacterial infections, immunology, and then we had parasitology, and it really just really interested me. I like the fact that the symbiotic relationship between them, how the parasites don't want to kill us because they live off us and all this kind of stuff. I just found it very fascinating. So my undergrad project was actually on cancer, but then I did a master's at York Uni as well, and did 2 projects there, one on malaria and one on leishmania, which is a skin-eating parasite, basically. And I wanted to carry on, but so the leishmania and African sleeping sickness are what's termed neglected tropical diseases, and they're called that because they're neglected on the fundraising side. There's not much money in them, so trying to get a PhD in them is quite difficult.
Yeah.
So I wanted to stay in health and disease, and then went into the PhD because it was all on wound healing. And then my department was the cutaneous department, the skin department, basically. So then over the 4 years I've been in that department, going to conferences on dermatology and going to the seminars every week that we had, I just became very well versed in skin biology, I guess. I was kind of a bit of an outsider in the group because I worked on the gingival tissue as well. So I have connections with dentistry, which luckily was next door. So it was quite handy to have contacts and all that kind of stuff.
Yeah. What drew you to biology in the first place?
So I did maths, biology, chemistry and further maths for A-level. That was a bit of a nerd. And I actually only got a B in, that's just quite a story, I only got a B in biology and got an A in maths and further maths. So you'd think I'd want to do maths, but it just wasn't really for me. And then I applied to go to Durham in St Andrews, and I didn't get the grades, so I ended up actually going through clearing to university, which might be quite good for anyone who ends up in clearing. I've still got a PhD from a Russell Group University in biology, so going through clearing is not necessarily the end all. I know plenty of people who went through it and ended up at very good universities with higher degrees. So I did biology just because it was the one that I was interested in. Chemistry was too hard. And biology just interested me. I come from quite a sciencey family as well. Both my brothers are doctors. One's a PhD, one's a medical doctor, and my parents are both scientists as well. So although I spend most of my time reading history outside of work, but I think I was a bit pushed towards the science side in the family.
So how have you brought your research to the public?
So I've done a few different things. I think public outreach is quite important, which is why I'm on the podcast. But I think one of the oddest things I've done is I wrote a poem and then did a stand-up comedy event. Wow. I also entered the poem into a couple of writing prizes as well. But it was one of those things where I got special commendations because people liked different aspect of it in the jokes, but I never won, which is a bit annoying. I think they were like, we can't really put this out as like the winning prize, but it was quite funny. So and
you're going to read some for us now?
Yeah, I could do. So this is the poem. It's talking all about the difference between wound healing, between gingiva and skin. I tried to make it a bit funny, but it's probably not. So now I'm going to talk to you about wound healing, something that sounds really appealing. I'll explore the differences between skin and gums and give details of my research outcomes. Now, the problem with healing of tissue cutaneous is that it isn't very spontaneous. However, this isn't such a big issue in and around the gingival tissue. The oral mucosa heals much faster, really showing skin who is the master. Furthermore, skin leaves nasty scars, while healed gums give the smile of Hollywood stars. There are several reasons for this difference, and I apologise for the lack of reference, but saliva, blood vessels, and inflammation, all of these provide important information. To study this, I build a 3D representation grown at the air-liquid interface to aid differentiation. These models are grown for 14 days, and tissue modelling, they're quite the craze. Firstly, the matrix structure is a mast made of collagen and fibroblasts. Then you add the keratinocytes to bring your research to new heights. Here, as you can see, there is a sample, which I hope proves to be a great example. I've stained for markers to show its structure. When they appear correctly, it brings me great fructure. Now, I know you're thinking of fructure's definition. It means when things come to fruition. It's an obscure word as fructure, but it's bloody difficult to rhyme with structure. Once I'd optimised the 3D protocol, next was the wounding for me to install. Using a biopsy punch to remove its centre, I created a wound for the cells to enter. After fixing the models, I can assess where the cells have come to rest. To improve this, it could be wise to use cellular fluorescent dyes. After all, it would be sublime to track the cells in real time. Then I can really put to rest whether skins or gums heal best. Therefore, thank you all for listening and hope you leave with smiles glistening, knowing a bit more about my project pieces after this, my 3-minute thesis.
Yay!
Yeah.
Cool. Shame that it's that the funding's not there for it to carry on. And so then it's just making me wonder then because it's a really good project, it's a good change, and I think it would be a life-changing thing. So if someone has their own funds, has a lot of money, and is interested in science, could they fund their own PhD? Is that possible?
Yes. I know a few people who've funded their own PhD. So basically, to fund your own PhD, you need to pay the university tuition fees. A lot of the time, you'd have to pay for your lab space, like rental, I suppose. And then, obviously, all your lab expenses on top of that, which in science can be huge, especially in the scoring area. So it's definitely doable. I think I know a few people who've done it, but it does add up. You would have to be quite rich to do it. Right. But then that's the beauty of it, because then you can do what you want, as long as you have your... So basically with the PhD, when you hand in your thesis has to be three things. It has to be novel, it has to show that you've done the work, and it has to fit into the wider scientific literature. And so as long as your project hits all those three things when you finish, there's no reason that you wouldn't be able to fund it yourself.
So how important is it to network at conferences or seminars that you go to?
I think it's really important. So, like I say, I've just finished my PhD, so at the moment I'm job hunting, and I'm looking for postdocs or academia, or going into industry and the like. But I found, although I have got a few interviews with people I haven't met, I found most of my opportunities have actually come from people I've interacted with at conferences or at seminars. So much so that I presented at the London Skin Club a while back, and at the end said I was looking for jobs, and had numerous people approach me. And I think when you go to conferences, it can be quite easy, and I've done it myself. If you go with your friends from the lab, you get a free dinner and you get a free drinks reception, and it can be quite easy just to hang out with your friends. I think actually going and talking to people and making these contacts and talking about your research is really important. Not just for the publication, as we were talking about earlier, the collaboration, but the opportunities that will give you in your future career.
Thank you very much for your time. Thank you to everybody for listening. We've got links up to our website, which is the Anti-Waffle podcast.wordpress.com, and our Twitter account, which is @AntiWafflePod, and we'll put links up to Chris's paper when it's published. Please do get in touch, as we'd love to hear from you. Bye.
Bye.
Bye.